All right, today's topic will be about secondary messengers, and it's about the steps that come from the ligand-receptor interactions and then turning them into a physiological response. But we'll specifically be highlighting the secondary messenger synthesis and what those molecules and chemical signaling molecules do for the body. So this is part of the AP Biology Standard 3D, that cells communicate by generating, transmitting, and receiving chemical signals. The primary learning target will be that students will be able to describe the signaling pathway from specific receptor-ligand interactions and the eventual biosynthesis of secondary messengers that then eventually facilitate a specific cellular response. So a brief overview about signal transduction in general. The reason signal transduction occurs is because we need to transfer some type of information into some other type of information. So for example, everybody has a cell phone. Your cell phone is specifically linked to a specific wavelength of radio frequencies where the radio signal will actually come in, perhaps from a phone call. So somebody's calling you. That radio frequency will hit your phone, and then it will be converted into a sound, so that you will actually respond to the phone and then pick it up and eventually talk, and that radio wave will be sent back. It's just an exchange of different information. So it's from radio frequency to sound so that we can actually respond to it. The same thing happens with cells. As you can see here from Albert's textbook of cell biology, there are very specific ligand, which is the signal here, and receptor interactions to each its own to facilitate some sort of intracellular response. And we'll get into that more in detail in this next slide. This is the sort of step-by-step process that will occur that has just these pretty little arrows. We'll get into detail with that in a little bit. But the primary thing I want you to know is that signal molecule, we'll say this is the radio wave, attaches to receptor, so phone, and then it goes into in the cell, and it also gets converted into a different type of signal, potentially to go out of the cell itself. So that could be potentially the sound that you hear, as well as the sound that you give back if the signaling molecule is going out. Now, why are secondary messengers important in general? Before we get into it, the thing that makes secondary messengers iconic, I suppose, in cellular biology is that they amplify a response to a stimulus. So not only does it make a response occur, but it makes that response occur quickly, efficiently, and very, very strongly. So for example, in fight or flight, secondary messengers are a big deal in that where an epinephrine binds and cortisol binds to receptors. It creates thousands and thousands of these secondary messengers to flood the cell full of activated enzymes so that the response can be mounted. Now, it also is like a kind of like army. So in the army, there's a lot of the times a spy. So that spy goes out and figures out some sort of information, goes back and recruits hundreds and hundreds of troops, potentially like there's a threat coming, and tells them, hey, we need to attack this crew. So they all of a sudden send thousands of troops into this one area based on one piece of information. That's very similar to the spy being the signaling molecule and the secondary messengers being that big mounted response like the army. Now, G protein coupled receptors are the primary beginning of this whole process. So as I've said, a receptor is bound to the transmembrane. I guess it's bound within the cell membrane, and this specific one, this GPCR, G protein coupled receptors, is made of two different units. Here, I'll show you on the next slide real quick. So here's this first unit. It's a receptor protein, and then there will also be an intracellular protein that is called the G protein. So here's the receptor, here's the G protein. As you can see, there's 7 different loops within this GPCR. There's also a binding site for the ligand or signaling molecule, like a hormone or polypeptide or sugar, or light energy, which we'll talk about here in a second. And there's also an active enzymatic area where it will actually activate that G protein that we saw in the last slide. Now, as I mentioned, this is the beginning stages of secondary messenger synthesis. This is the initial information that is received by the cell. And one really cool thing that can happen is actually in bacteria, you can study this on your own. This is in the reference page of the very last slide. Bacteria, some archaea specifically, have a protein, a G protein, called bacteriorhodopsin. And it actually binds to a photon, which is a unit of light, or a little pigment of light. And as it hits that specific enzyme, it actually pumps hydrogen ions into a lumen, creating a chemical gradient, which eventually powers ATP synthesis. So these bacteria can actually live almost like plants, where they're creating energy, not in the form of sugar, but in the form of ATP, simply by using light and converting it into a chemical gradient. Now, let's talk a little bit more about the activation of these G proteins. Once a G protein coupled receptor actually binds with a receptor, so as we can see, or with a signaling molecule binds with the receptor and activates it. So anytime something binds in biology, some sort of conformational change, so a physical change happens in that receptor or in that thing, and then it does something else. So for this case, the signaling molecule binds to the binding site, which activates this activated domain of the receptor, thus binding to a G protein, and it actually dissociates GDP, so guanosine diphosphate, into GTP, which activates the alpha subunit. It's a 3-subunit protein, the alpha subunit of the G protein. And it also, in some cases, activates this beta-gamma complex, so the second and third units. This is a trimer protein because that's 3 different units. Now, since this binds with GTP and since this is activated as well, these things will go on to phosphorylate or activate other proteins and enzymes in this cycle. So let's go to that. Now we're getting into the synthesis of the secondary messengers. I will talk mostly about CAMP, cyclic AMP, as well as briefly about diacylglycerol and calcium ions. But let's look at this model here real quick. As you can see, adenyl cyclase binds to this alpha unit of the G protein that was just activated by the receptor. And this is a really important enzyme, adenyl cyclase, because it kind of breaks down. It takes 2 phosphates away from ATP and circulates them. So it actually creates cyclic AMP, adenosine monophosphate. And this is our secondary messenger. This secondary messenger, hundreds of them will be made, which activate pKa, a kinase, and this will eventually go through a few different steps to finally create the physiological response of glycogen breakdown. So a lot of these signaling molecules, these secondary messengers, activate different kinases or phosphorylases to activate other enzymes to eventually break down glycogen, to eventually stimulate inflammatory responses, a lot of different things. So for CAMP, we'll talk about that a little bit. Diacylglycerol is one that is broken down into 2 different subunits, and then it increases kinase affinity to calcium ions, which then activates those kinases. Specifically, in this one, I believe it had to do with not hormone secretion, but vasodilation. Furthermore, calcium ions are also used in sea urchins. So calcium ions themselves can be secondary messengers. And this happens in response to fertilization in sea urchin embryos. And it's the slow block to polyspermy. So you can look at this in the references again, where calcium ions are used as a secondary messenger, getting that response that, hey, we just got fertilized. We don't want more sperm to get into this embryo. And then it actually increases the vitelline envelope, thus pushing any other sperm away. So that's another fascinating thing that secondary messengers do. But let's talk a little bit more about cyclic AMP. Here's one specific example where cyclic AMP is actually changing gene expression. So once it activates pKa, this turns into a nuclear receptor where it can actually go into the nucleus, bypassing this double membrane of the nucleus, binding to a transcription regulator, which was first inactive, but now it's activated by pKa. And this will bind to different promoter sequences of genes to increase their expression. So for example, when testosterone binds to receptor, it eventually ends in the upregulation of different proteins that have to do with like hair growth in men, as well as deeper voices and larger muscle mass, increased body odor, etc. So that's all amplified by cyclic AMP. Now, there's so much more that cyclic AMP and many other secondary messengers do. However, if you want to dive more into this, you can look into this Serrazani et al. This has a lot of really cool physiological changes in response to secondary messengers, a couple of them being cholera and pertussis. Pertussis causes whooping cough in infants. And in cholera, if you look at this model over here, in cholera actually amplifies the G protein coupled receptor, and it doesn't turn it off. So another big thing with cell biology is anything that's turned on must be eventually turned off in order to maintain homeostasis. However, in cholera amplifies this G protein coupled receptor, making the alpha subunit bind to GTP, thus activating adenylyl cyclase. And all of these secondary messengers are constantly being made, which increases inflammation, which causes diarrhea and a lot of other bad symptoms of cholera. And pertussis, it's the opposite. Pertussis actually blocks this trimer of the G protein coupled receptor from dissociating. Therefore, this cannot occur. So adenylyl cyclase does not bind to ATP, does not make cyclic AMP. So no immune responses can be mounted, no inflammatory responses that are healthy can be mounted, so it actually suppresses your immune system at a greater rate. And as I said, this website can also show you different changes in smokers and COPD and how those manifestations of symptoms can actually affect your cellular biology, thus inhibiting or promoting too much of cyclic AMP and other secondary messengers. A few other resources that you can check out in response to secondary messengers. They're actually linked to major behavioral disorders such as bipolar and manic depressive. This is because that these secondary messenger cascades are being amplified, over-amplified, so you get a stronger stress reaction at certain times, and then other times it's down-regulated, so that's where that manic depressive state comes in. And there's actually some drugs, and I believe lithium is actually another part of that resource that talks about blocking those different pathways, just as cholera, and here I'll show you, just as cholera did in the last slide, or pertussis, sorry, just as pertussis blocked this pathway, it's very similar with these drugs. They block certain pathways so that these secondary messengers can be diminished, therefore creating a better life for the people experiencing these things. And finally, there's also cyclic AMP and pKa pathways that are linked to osteogenesis. So osteogenesis is the creation of bone tissue. And for people who struggle with osteoporosis or osteopenia, so brittle bone syndrome with low bone density, these different pathways can be linked directly to osteogenesis. So drugs, hopefully, can amplify these messengers up a bit so that these, osteogenesis occurs when CAMP and pKa pathways are actually diminished. You'll read this in the source. So drugs can help diminish this cyclic AMP response, therefore promoting bone growth. A lot of checks and balances here. There's a lot of promoting, a lot of repressing, and all of them are linked to keeping organism homeostasis. So as you can see here in the end, there are a lot of different resources you can check out. I highly recommend you look at each and every one of them. If you're ever confused, go back to your Essential Cell Biology textbook from Alberts. Chapter 16 deals with all the cell signaling, gives very clear-cut details about how these things occur. And these will be more of that deep dive, in-depth information that you can check out at your leisure. If you have any questions, email me. Thank you for your time and have a great rest of your day.